my name is Blair Burlingame, and this is my anatomy project on advancements in medical technology. Advancements in medicine are made daily and are vital to new research, new cures, and new methods being discovered. These are then used in order to treat diseases, heal injuries, and make our bodies a little healthier. Today, we will be looking at recent medical advancements that are associated with the integumentary system, skeletal system, muscular system, and nervous system. First, we will start by looking at the integumentary system. A recent medical breakthrough for the integumentary system is better skin grafts for healing chronic wounds. This was found in June of 2017 by Dr. Jeffrey Litt at the University of Missouri-Columbia. The problem with current skin grafts is that more than 6 million cases of chronic wounds occur each year and cost $20 billion each year in the United States. Diabetic ulcers, pressure sores, surgical site wounds, and traumatic injuries account for most of the wounds that won't heal. However, in the study, Litt's team used a recently developed autograft harvesting system to care for patients with various types of chronic wounds. The new technology was used to harvest only the top layer of the skin, known as the epidermis, for a much smaller, consistently sized donor graft. The minimally invasive approach resulted in much less donor site damage and little to no pain following the patient's care. Clinical outcomes and costs were evaluated. This resulted in a much faster healing of the wounds, much less pain on the patient, and a reduction of cost of $1,100 per case. Next is the muscular system. This has to do with scientists getting one step closer to being able to create three-dimensional polymer structures that closely resemble human tissues. This would give scientists and medical researchers the ability to create artificial organs, and this study is specifically working to being able to create an artificial heart. An artificial heart would be an absolute lifesaver for people with cardiac failure. However, to recreate the complex organ in the laboratory, one would first need to work out how to grow multi-layered living tissues. Researchers have now come one step closer to this goal. By means of a spraying process, they've created a functioning muscle fiber. This was done in November of 2017 by the Swiss Federal Laboratories for Material Science and Technology in Zurich by Lukas Windenbacher and his team. He explained that until now, the culturing of multi-layered functional tissues has been a major challenge in the up-and-coming area of tissue engineering. However, researchers have now succeeded in letting cells develop into muscle fibers in a three-dimensional synthetic polymer scaffold. This is done by a process called electrospraying. Basically, spraying the cells out of a spray can in layers to give them time to bond and morph together into a tissue. During this process, gossamer-like threads of liquid polymer are interlaced in the manner of natural tissue. Then, the special spraying process makes it possible to inject the capsules into the pores of the spun scaffold. Scanning electron microscope images show that the cells feel at home in their synthetic polymer nest. As soon as the capsules have dissolved, the immature precursor cells begin to join up and to mature to form elongated muscle fibers. The aim is to end up with a structure that resembles natural muscle tissue as closely as possible. While this still is a long way to go before becoming a full functioning heart, this is one step closer for scientists to build upon. Next is the skeletal system. For this, we will be looking at our most precious members of society, newborn babies, specifically premature babies. New studies show that an increased amount of vitamin D given to premature babies increases the growth of their bone density to allow them to develop and stabilize faster. This data was discovered in October of 2017 by Dr. Ann Anderson Berry at the University of Nebraska Medical Center. She specifically found that the standard supplementation of 400 IUs of vitamin D would be increased to 800 IUs daily. With this done, there are reductions in the number of premature babies and preterm babies with extremely low bone density. This gives these babies' bones a quicker start and a little bit of a kick in order to come into this new world and adjust properly. Finally, we look at the nervous system. New research has emerged that changes the long-standing ideas about how the axons grow during embryonic development. The findings of the study could help scientists replicate or control the way axons grow, which may be applicable for diseases that affect the nervous system, such as diabetes, as well as injuries that sever nerves. This was found in April of 2017 at the Eli and Edith Broad Center of Regenerative Medicine and Stem Cell Research at UCLA by Dr. Samantha Butler. As an embryo grows, neurons extend axons into the developing spinal cord. Axons are then guided to reach the other areas of the body, such as the brain, to establish a functioning nervous system. It has been generally understood that various guidance cues, which are cellular molecules such as proteins, either attract or repel axon growth as the axons reach out from the neurons to find their destination in the nervous system. Previous research suggests that a particular guidance cue, called neurotron 1, functions over a long distance to attract that organized axon growth, similar to how a lighthouse sends out a signal to a ship from afar. However, previous research also shows that neutron 1 is produced in many places in the embryonic spinal cord, raising questions whether it really acts over a long distance. Most notably, neutron 1 is produced by a specific tissue stem cell called neuroprogenitors, which can create any cell type in the nervous system. Yet, it was not understood how neutron 1 produced by neuroprogenitors influenced axon growth. Butler and her research team removed neutron 1 from neuroprogenitors in areas of mouse embryonic spinal cords. This manipulation resulted in highly disorganized and abnormal axon growth, giving the researchers a very detailed view of how neutron 1 produced by neuroprogenitors influences axons in the developing nervous system. They found that neuroprogenitors organize axon growth by producing a pathway of neutron 1 that directs axons only in their local environment and not over long distance. This pathway of neutron 1 acts as a sticky surface that encourages axon growth in the direction that form a normal functioning nervous system. Butler's study is a significant reinterpretation of the role of neutron 1 in nervous system formation. The results further scientists' understanding of the contribution neuroprogenitors make to the neural circuit formation. Determining how neutron 1 specifically influences axon growth could help scientists use neutron 1 to regenerate axons more effectively in patients whose nerves have been damaged. For example, because nerves grow in channels, there's much interest in trying to restore nerve channels after an injury that results in severed nerves, which is often seen in patients who've experienced an accident or in veterans with injuries to their arms or legs. One promising approach is to implant artificial nerve channels into a person with a nerve injury by giving regenerating axons a conduit to grow through. Butler believes that coating such nerve channels with neutron 1 could further encourage axon regrowth. Her continued research will focus on uncovering more details about how neutron 1 functions and how it could be used clinically. Scientific advancements are only a small step in the cascading effect that their knowledge could lend. We could better treat diseases, heal injuries, and make our bodies a little healthier. Thank you and have a great day.